In this video, we're going to take a look at a large family of proteins, which are what we call your enzymes. Now, enzymes play a very, very critical role in digestion of nutrients, and so it's important not just to understand their structure, but more importantly, their function and how they do what they do, or at least the basics of such. Now, enzymes, if we classify them from a chemistry perspective, are what we call your body's biological catalysts. Now, a catalyst is any chemical that can accelerate a chemical reaction, making it happen faster while not actually being consumed by the reaction itself. So if we think about digestive enzymes, for example, remember that the goal of a digestive enzyme is to take polymers, the form of nutrients that are found within your food, and break them down into monomers so that they can more easily be absorbed from your digestive system into your blood and ultimately transferred to cells. This means that it's an enzyme's job in order to break down polymers into monomers, which is why when we write a chemical reaction like this, the enzyme doesn't go on the reactant side or the product side, because it is involved in the actual process itself rather than being broken down or produced in the chemical reaction. This also means that your body's enzymes are reusable, meaning your body doesn't need to keep making new enzymes every time it needs to break down nutrient polymers. Now, this particular enzyme is an enzyme that is found in the liver, and it is an enzyme that we call catalase. We use this enzyme as an example because you can actually do this experiment from home. All that you need to acquire is a sample of pork or beef liver that you can get at most butcher shops around. And the job of catalase is not quite as a digestive enzyme, but this is a detoxification enzyme, meaning that it is used to break down a toxic chemical known as H2O2 or hydrogen peroxide, which is a chemical that we use as a disinfectant, and you can buy this at any grocery store. Now, the way that an enzyme works is that H2O2 in this specific case is a substance that we call a substrate, and a substrate is any substance that is actually able to enter the enzyme and have the enzyme perform a chemical reaction on it. The substrate is able to enter this enzyme because of the most important part of the enzyme, which is what we call the active site. In the case of catalase, the active site is right here. And when the substrate enters the active site, the enzyme does something in order to actually alter the chemical structure of the substrate that entered. This is the process that we call catalysis, where the enzyme actually performs the chemical reaction to cause a polymer to break down into a monomer, or in the case of this toxic substance here, catalase is going to break down hydrogen peroxide into two chemicals that are no longer harmful to the body, water and oxygen. So we can see here that even though this is not a digestive enzyme, it performs a similar function by taking a larger substance and helping it to decompose into two smaller, less harmful substances. Now, the truly cool thing about enzymes is that each enzyme has a truly unique active site, meaning that each individual enzyme will only fit one specific substrate into its active site, simply because each individual substrate has a specific shape or a specific chemical charge, and if neither of these matches the active site, the substrate is not able to enter. And this is what makes enzymes so specific, because each enzyme only has the ability to speed up a single chemical reaction based on the ability of a specific substrate to fit within the active site. So what exactly happens when we look at what happens in the active site? Well, here, let's take a look at an actual hydrolysis reaction. So in this case, we're going to be looking at a carbohydrate here, and our goal of this particular enzyme is to take this carbohydrate, which is made of one sugar here and another sugar here. Each of these are carbohydrate monomers, and this enzyme is going to break the two monomers into two separate molecules like this. So here we see that the enzyme substrate, or the enzyme's active site rather, does not match the substrate entirely. We can see that these sugar molecules have a very specific shape, and they do not exactly match the shape of the active site, and this is deliberate. However, the active site, due to the charges that it has relative to the charges of the individual molecules in the substrate, the active site can still attract the substrate and cause it to enter the active site, but due to the fact that the active site doesn't match exactly, the active site is going to basically force the substrate into it, therefore making the substrate molecule more unstable, which ultimately causes it to break. So in this simplified diagram here, we can show the sides of the active site are kind of compressing the substrate together, causing this bond here to become more and more unstable. Now, remember that enzymes have the role, or digestive enzymes rather, catalyze what we call hydrolysis reactions, meaning that once this bond is unstable, a water molecule can then be added in order to break that bond that is holding the two monomers together into separate monomers that can then be released from the active site. So here we can see the individual monomers make up the products of this chemical reaction, and now these monomers are released from the enzyme's active site. And now we can see, after the monomers have been released, that the active site is now empty, and this allows the enzyme to be reused to do the reaction all over again. And this means that as long as your body is supplying carbohydrates for this enzyme to break down, this enzyme can theoretically be used an unlimited number of times as long as it doesn't take damage in the process of digestion. Now, because of the fact that the sugar molecule, in this case, the substrate, does not actually match the active site completely, this is what causes the enzyme to actually force its will, so to speak, on the substrate, which is why we call this model of enzyme activity induced fit. Remember that when you induce something to happen, the process is not completely voluntary. Some people would use the verb to force instead of induce. I prefer a more subtle verb, the verb to compel. So again, because the substrate does not match the active site 100%, the instability of the substrate and active site compels the substrate to break when the enzyme tries to force the substrate into the active site like this. Now, if we take a look at an actual reaction that's going on here, this is actually the reaction that goes on inside of the mouth. So this is a digestive enzyme that we call amylase that breaks down starch as soon as it enters your saliva. This enzyme is present within your saliva, and once again, starch has the ability to fit within the active site of the enzyme, but the structure of starch does not match the active site completely, and so it will need to be forced in, where the enzyme induces it to break. And once each of these bonds is made unstable, that means that a water molecule from your saliva can be added in order to break the bond that holds each individual sugar monomer together. And in the end, what we would have is a whole bunch of individual carbohydrate monomers that can then be absorbed into your bloodstream and transported throughout the body. In the follow-up to this video, we're going to look at several environmental factors, most notably temperature and changing acidic or basic conditions and the consequences that changing the environment has on the structure of proteins, and therefore how enzymes will respond once their environment has been altered.